necessary.
There was a whole discussion with the speakers whether, because we're more than 6 feet apart, right? So in theory, whether
Yeah, no, the GSA, we asked before, like before the meeting, way before the meeting, we said, come on, can just the speaker take off their mask? Right. And Susie said that that was an emphatic no from the GSA.
But that she wasn't going to drag people up the stage.
oh, well, there you go. So that, I think that some people want to go rogue.
Hey, Erica.
Hi.
Between each one, just push the button here, it'll reset.
Oh, should I start or you?
Okay, so I push this between.
Yeah.
And then, but there's that little floating thing. Is that going to?
Yes.
You go into that,
Uh-huh.
and then from there, there's something that says turn off.
Okay, but we have to hit refresh between every talk? Okay. I like that it's a big button to smash. To smash.
It's magic.
Are you nervous or are you just one of these people who's called?
I'm super nervous, and then
You get in the zone.
so and like I had
No.
really nightmares about my talk, like
But-
like. No. But it's fine. I had nightmares. It's fine. It's fine. It's okay. People should know I'm nervous.
All right, go away so I can introduce you. Sit down somewhere. Yeah. All right, good morning. Oh, that is loud. Good morning. We're going to get started with our final plenary session. And our first speaker is Guy Tanentzapf from the University of British Columbia.
All right. First, I'd like to thank the organizers. This is, for me personally, the most meaningful invitation I've ever gotten because you guys are my science family, so it really means a lot to me. And I thought it's morning, so I thought we could start by some jazz hands, some stretches. Can I get some stretches? All right. So I just coordinated the behavior of a group of idiosyncratic individuals in this room. There's somebody in Australia, in India, and in Israel right now going like this, and people in their roommates are looking at them like they lost their minds. And the question of how cells, because our cells in our bodies and the bodies of the fruit flies we love, undergo this sort of coordination in space and time all the time in complex organs. And that's really what I'm going to talk about, is how you coordinate behavior like this without a microphone anyways. And the way we've been thinking about this is, at least I've been thinking about this, is in an analogy to this incredible biological phenomena of flocking or murmuration. And much like starlings, hundreds of thousands of starlings in this flock of birds, cells exist as independent agents in an organ. They act collectively. They interact locally with one another and the environment. And the sum of these local interactions between cells lead to the emergence of global behavior. And we, in particular, the long-term interest in my lab is in studying cell junctions. And we want to understand how cell junctions allow cells to interact locally with one another and with their environment. And these are all the type of junctions that we are currently studying in my lab, but I'm sure we are happy to extend it. We'd love cell junctions. So this is an overview of my talk. I'm going to start by introducing the overarching question, which I already alluded to. I will introduce you to the model system we use, and then I will tell you 3 vignettes. So these are parts of stories that I feel help us go towards the goal of answering our overarching question about ECM, about gap junctions, and about the power of live imaging. And then, time permitting, I will go on about wild speculation. So stick around for the wild speculation. And as I already introduced, the overarching question, which is what mechanisms coordinate cell behavior in vivo across large distances and time. And the hypothesis that drives our work is that, and I hope to convince you too, is that cell junction-based mechanisms coordinate cell signals in space and time. And the particular decisions that we are interested in are the one that that is related to the regulation of stem cells. So when a stem cell divides, the daughter cells have a very important decision to make. They can either make another stem cell, they can self-renew, or they can differentiate. And getting this decision is really important because if you do make too many stem cells, you can end up with cancer. If you make too many differentiated cells, you deplete the stem cell population, and you end up in premature aging or tissue degeneration. And most stem cells, this is a really important decision. So most stem cells need help to make this decision. And this help is provided by a stem cell niche, which is a signaling microenvironment that provides specific cues that controls the behavior of the stem cells and helps them make the right decision. And the particular model system we use that I'm going to talk about today, we use a number of model systems to study this in my lab, but I'm going to talk about the lymph gland. The lymph gland is a larval organ that is the main site of the production of the blood cells that mediate the larval immune response. It is found near the head, attached to the larval Drosophila heart, and it is mostly quiescent, but it can be activated upon infection or infestation by wasps, for example, and ends up creating 3 different types of mature blood cells that are very different and specialized: the plasmadocytes, the lamellocytes, and the crystal cells. And the lymph gland has a beautifully simple structure. It has a posterior signaling center, which is the hematopoietic niche at one end, which is shown here in green in both live picture and the schematic. It has a medullary zone, which is where the stem, a large medullary zone where the stem or progenitor cells are found. And it has a cortical zone, which is the shell on the outside that contains the differentiated mature blood cells. And what I want to emphasize is that architecturally, it's a pretty complicated large organ that has a large number of progenitors, hundreds of progenitors surrounded by this shell of differentiated cells. And importantly, look at how far the stem cell niche, which provides instruction, is from these progenitors. Some are really close, some are really far, and yet their behavior is highly coordinated. In addition, the point of this slide is to illustrate the complexity of the signaling environment in the lymph gland. So I wanted to make this slide confusing. So if you're confused, that's good, it's working. What it's supposed to show you is that virtually every single signaling pathway that is known to work in flies at some point is doing something in the lymph gland. DPP, insulin TOR, Notch, Hedgehog, Wingless, JAK-STAT. If you discover a new signaling pathway, look in the lymph gland because it's going to be doing something. So you have this incredibly complicated signaling environment in addition to this very complicated architectural environment. And yet it's, they're very coordinated in their behavior, the progenitor cells. So our first vignette to help us understand how this happens is dealing with how the ECM helps to spatially coordinate cell signaling in the lymph gland. So here is, this is something that was noticed already before we got involved, but we explore this further. And this is a 3 dimensional reconstruction of the ECM environment in the lymph gland. And you can see that the big green tube is the heart, and everything around it is the lymph gland. And you can see how the progenitors are ensconced within this complex, beautiful extracellular matrix. And this is exciting for us because my lab, one of our main focus for a long time is about studying how cells interact with the ECM. In particular, we focus on integrins, which are the main molecules that mediate the attachments of cell to the ECM. Integrins are a heterodimer of an alpha and beta subunit. They bind to the ECM extracellularly and then span the membrane and assemble a large signaling and adhesion complex inside the cell that connects to the cytoskeleton. So what we did is we knocked down integrins, in this case, both different integrin subunits in the lymph gland, and we see that both the maintenance of the blood progenitors is disrupted. They are not maintained very well. And we see a huge increase in differentiation of mature blood cells. So the maintenance of the progenitor depends on ECM adhesion through the integrins. So what are the integrins and the ECM doing in the lymph gland? Why are they important for the maintenance of the progenitor cells? And the hypothesis is that they anchor cells or stem cells or progenitor cells in space. So what we did is we developed a technique that I will go on and tell you a little bit more about, of long-term ex vivo culture of the entire intact lymph gland. And what we see is that if we look over a course of many hours, we do not see, either in a wild type or in an integrin knockdown, any movement of the progenitor cells. They are maintained in space. This is not, the role of integrins is not to maintain them in space. So they're doing something else. What are they doing? So when we did this work, what we noticed is that if compared to a wild type, which is shown here, when you knock down integrins, you have a large reduction, which is quantified here, in the density of the extracellular matrix, okay? So that brings rise to the question of, what are the implications of this? And what we find is that there's a correlation between the density of the ECM and the maintenance of the progenitor pool. So if we reduce the density of the ECM, in this case through a collagen mutant, we find a large decrease in the maintenance of the progenitors and an increase in differentiation. So density is important, okay? So why does the modulation of ECM density affect blood progenitor maintenance? And the answer that I will show in the next couple of slides is that ECM density is a universal regulator of multiple signaling pathways in the lymph gland. So this is again building on previous work, which is cited here. And what we found, we extended this work, is that when you lose integrins, you have reduction in the ECM density, you see just a general disruption to multiple signaling pathways, in this case, DPP signaling, wingless signaling, and hedgehog signaling, and they are universally disrupted in the lymph gland when we have change in the density of the ECM. So why is that? So imagine there's a signal that comes, there's a stem cell niche and it produces some sort of signaling that moves through the space of the lymph gland. Now, if you throw an ECM on top of that, that regulates how far the signal can move, and also its distribution in space in a universal way throughout the lymph gland. And as you can imagine, if you increase the density or reduce the density of the ECM, that affects the distance that the signal can move, how it's distributed throughout space, and that will change the behavior and the outcome throughout the lymph gland. So the model from the first part, my first vignette, is that cell ECM junctions are general modulators of cell communication that act on multiple pathways by altering the density of the extracellular matrix. Is this regulatory mechanism physiologically relevant? So what we find is that when you infect flies with bacteria, for example, we see a general disassembly of the ECM. The ECM density becomes less. And as a result, you see the characteristic increase in differentiation and the reduced maintenance of the progenitor pool. If you block this, so we have mutations that block this reduction, that increase the density of the ECM, you block the initiation of the immune response. You do not make most stem cells, the progenitors are maintained just fine. So density is an important regulator, ECM density is an important regulator of the modulation of the mediation of the immune response. So that brings us to our second vignette, which is how gap junctions help temporally coordinate cell signaling across large distances in the lymph gland. And this again builds on some of the beautiful work in the field from Jiwon Shim working in Utpal Banerjee's lab, who showed that calcium signaling activity is both a positive and a negative regulator of blood progenitor behavior in the lymph gland and the production of mature blood cells. So we extended this technique that we have. We dissect the lymph gland and can culture them for up to a day in images throughout this time. And in this case, Kevin Ho is a very talented graduate student in the lab, was expressing the calcium signaling marker GCAMP that detects calcium signaling in the lymph gland. And what we see is robust calcium signaling in the gland, which is shown here. So you see a lot of these flashing lights telling us there's calcium signaling. And when we look a bit closer, we start seeing things that look like this. So what we have here is a wave of calcium signals that travels from one cell to another. And we see calcium waves traveling great distances in the lymph gland, 10 cell diameters. They can go very far. And what that allowed us to do is look whether sometimes where the cells coordinate these calcium spikes, and use that to say how likely is this to happen compared to just random chance that would happen, and assemble something called a cross-correlation network. So this tells us that the behavior, the calcium signaling in these cells that can be very distant is coordinated. So we see coordinated signaling behavior. So the question is, how is this network formed? And what mediates the exchange of calcium signals between blood stem cells? And the hypothesis is that calcium signals travel between cells through gap junctions. So my lab has had also a long-standing interest in gap junctions, and shown here is that one of the gap junction proteins in Drosophila, called innexins. So here you see that at least one innexin is expressed in the lymph gland. Gap junctions are protein complexes that form a pore in a cell. They dock to equivalent complex in a neighboring cell, forming a channel allowing the passage of small molecules, nutrients, small signaling molecules, small peptides. So that's a good candidate. So to test that, we used the calcium blocking drug CBX. Our system allows us to add small molecules while we are to a culture. And we see that with in a dose-dependent manner, when we add CBX, we see loss of network connectivity within a cross-correlation network, and a strong reduction in these calcium propagation events, these waves that are traveling through the progenitor cells in the lymph gland. So this is illustrated, or we can also address this genetically. Here's a control, and this is what happens in a knockdown in the progenitors of a gap junction protein. In this case, one that's called innexin 4 or zero population growth. And this is a null mutant in innexin 4. And you can see that we basically lose this beautiful network that coordinates signaling behavior amongst the progenitors. So what is the functional consequence of losing gap junctions? I showed you we can knock them down genetically. So not surprisingly, we find that when we knock down gap junctions, we see a loss of the progenitor population and a drastic increase in the amount of mature blood cells that are produced. So is this regulatory mechanism physiologically relevant? Here, we see what happens when there's an infection, and we find that upon infection, we see a reduced number of progenitors. We see when you have an infection, you induce the production of mature blood cells. And again, as perhaps not surprisingly, we see a reduction in the amount of calcium signaling and the number of these propagation events. So one way to induce immune response, the production of immune cells, is to reduce calcium signaling and therefore reduce the coordination between the progenitors in the lymph gland. So I've showed you two ways of universally regulating the behavior of the progenitor in the lymph gland across time and space. One is by modulating the density of the ECM. The other one is through the formation of a network that connects the progenitors and allows gap junction mediated communication and exchange of calcium signals. What I want to talk now is how are we extending that work to look at how we can use long-term live imaging to learn more about this coordination and how we see how even small changes in single cell behavior can have large-scale impacts on the behavior. So this is more of a systems approach to the problem of coordination. So we can make movies. Our system is fantastic, and you'll see here, this is a lymph gland progenitors that are expressing a marker for progenitor fate, and here's they can divide, and this is a self-renewing division where you have two progenitors that are produced of equal size, and they express the same level of the progenitor marker. So this is a self-renewing division, which was not observed before in the lymph gland, based on what, as far as we can tell. And we are able to learn a lot about cell division patterns and a lot of other factors of a lot of other components, descriptional components of the behavior of these progenitors. But what I'm going to talk about specifically right now is about what we see when we study differentiation. So this is a progenitor cell that expresses a green marker for the progenitor fate. And this is a red marker for the differentiated cell fate, which is expressed in the nucleus. And you can see that with time, we see the general reduction in the amount of the progenitor marker and an increase in the amount of the differentiation marker. So essentially what we're watching here is differentiation in real time. And we can watch hundreds of these cells, and we do a lot of automated image analysis, and we can learn a lot about differentiation by watching it in real time. For example, one thing we can learn is we can look at the trajectory of differentiation. So this is sampling the same cell many, many times, looking at the amount of the progenitor marker and the differentiated marker, and we can see a transition from one state to an intermediate state to finally a state where there's high differentiation marker, low stem cell marker, low progenitor marker. So this is a particular trajectory of differentiation. However, this is maybe what's expected, where you go from a high progenitor marker, a low differentiated marker, to a high differentiated marker, low progenitor marker state. But we see another type of differentiation, which is shown here. And this is a cell that starts by expressing high levels of both the differentiated and the progenitor marker. And then the progenitor marker goes down, leaving us with only high levels of the differentiation marker. And this is a different type of differentiation that we see. And what you can do is this is another way of plotting that, where you see the differentiation marker in magenta and the progenitor marker in green. And this is the one where it starts with a high progenitor marker and it goes down, and you have an increase in the differentiation marker. This is the second type of differentiation, where both are high at the beginning and then the progenitor marker goes down. And we can plot to calculate, to look at this in terms of rate. We can calculate the ratio of one to another and plot it over time. And we get, for this type of differentiation, we see this characteristic sigmoid, what's called a sigmoid curve that has a slow phase and a fast phase. And for this type of differentiation, we see this linear curve. So that's what we call them. We have a linear trajectory and a sigmoid trajectory of differentiation. We see two different types of differentiation. So what is the physiological relevance of this observation? How is blood cell differentiation increased following infection? And what we find is that following infection, the individual rates of these different types of trajectory are not changed. So they both work equally fast. But, and we don't see any change in the proliferation. We actually see less proliferation of the progenitor pool. So if you're not making more cells and they're not differentiating any faster, how do you end up differentiating more cells, which you do? You get a lot more cells, mature blood cells being made following infection. And the way you do that is you bias the type of differentiation that takes place. You basically go from this number of cells that undergo linear trajectory to this number. So you see a massive increase in the pool of cells that undergo linear the linear differentiation trajectory, this is shown here, we bias it. And because we have more of these type of cells, we end up with more differentiated cells. So what this tells us is that all of these factors like gap junctions and cell ECM junctions, one way that they can mediate their effect of coordinating global universal behavior and regulate decision making on a global scale is, can be through even mild changes in proliferation rate, differentiation trajectory, differentiation rate, and proliferation or quiescence. And by regulating these factors even slightly, you can have an emergent phenomenon where suddenly you see much more differentiation or quiescence. So you don't have to have major changes, even small changes on a global scale can shift the balance in a particular way. So that's what one of the things we've learned through our live imaging, which brings me to the wild speculation that I promised you. So something I'm particularly very interested in is in systems theory. And what I would like to propose is that the regulation of hematopoiesis in flies exhibit features consistent with something called swarm intelligence. Swarm intelligence is a field of computation biology. Computation. It's big in AI. It has many applications. And swarm intelligence specifically deals with the collective behavior of a decentralized self-organized system, which is a little bit like what the progenitors are in the lymph gland, and in many other organs in the Drosophila body and the human body would be my proposal. And this population of individual agents follows simple rules. They interact locally with one another in the environment. And these local random interactions between agents or cells lead to the emergence of intelligent global behavior, much like the swarm, right? So we can use the computational tools and knowledge that is being developed to deal with a concept in swarm intelligence and apply it to how we coordinate behavior in the lymph gland and in many other organs in the fly body and the human body. And that is kind of my wild speculation general takeaway. I would like to conclude by thanking Kevin Ho, who's a wonderful graduate student who did all the gap junctions and was been very involved in live imaging and also helped with the ECM project. Rosalind Carr is an absolutely phenomenal undergraduate student in the lab who wrote most of the code to do the image analysis that I showed you today. So if there's any undergraduates out there, this is what you can do. You can make a huge contribution to our work. Rohan Kadilkar was a postdoc in the lab who's now an independent investigator in AceTrack in India, and he did all of the ECM work and made really nice, very invaluable contributions to how we think about these problems. And I know he's always recruiting, and he's an amazing person to work with. So I would like to conclude and take any questions. Thank you.
Celeste? Great talk, Guy. That was cool. So in the early part of your talk, you said that infection modifies the extracellular matrix, and that then changes the signaling pathways and differentiation. How does that work?
I don't know. I think there's a few avenues we can follow. Obviously, proteases are very strongly associated with infection. So that's an avenue we can pursue. But if anybody has any ideas, I'm open to them. I always tell my students the most important 3 words whenever you give a talk. I don't know. So I'm going to use that. I'm going to follow my own advice.
Lynn?
Hi, Guy. That was wonderful. Thank you. I loved looking at your kind of whole node live imaging. And I'm curious if you have the resolution in that system to look at the effect on cell divisions of modifying the ECM or the gap junctions. Can you observe divisions going on? And
yeah, that's a brilliant idea. Thank you. Yeah, we should do that. I'm not giving you authorship though, but maybe I should. But that's a really good idea. I mean, we just right now we have a paper that we're revising just to describe it. And there's a lot of controls. I mean, this is a fraction of a story, but for example, we look at whether the divisions are polarized. So there's many things. This was just to build a pipeline of analysis. And now we can apply it for things like ECM. And Kevin, and I know you're watching this, take notes. Yeah.
All right, Lucy.
Guy, that was incredibly exciting. Building on the ECM questions, it sounds like you don't know yet whether the sigmoidal differentiation trajectory correlates with being far away from ECM.
Yeah. Yeah, we can look at that. That's a good suggestion also, to look whether they, because we spend a lot of time, I mean, Rosalind did incredible work looking at where differentiation, she might even have this data. So we segmented into regions, the entire lymph gland in 3-dimensional, and we can see where differentiation takes place and where division takes place. So this is something we can look at when we might have this data. I just haven't thought about that, but that's,
Yeah, since they both.
yeah. So you're saying basically saying
Since
which part of which
In
type of
infection,
differentiation
you
occurs where.
lose ECM density, and then also in an infection, you get more sigmoidal trajectories.
Yeah,
Do you see de-differentiation?
we haven't seen that yet. We have not seen that yet,
Thank you.
as far as I know. Again, Kevin, take notes. Yeah.
Great talk, Guy, and yoga instruction. What do you know what's downstream of the calcium?
Yeah, we have this is published for this paper that came out last year, and we know there's CAM kinase, so we do have some of the downstream. And actually, what's really exciting for me is that we show that. I mean, this was a very simplified version of this. Essentially, when you change the connectivity, you change how calcium encoding takes place, right? So for example, you see a lot more spiking of the low amplitude spikes. So what you're changing is it's not like a yes, no kind of thing, which I presented it as just for simplicity. You actually change the type of calcium information that is encoded, right? And we have a theory how that might work, which is to deal with maintaining a certain level of CAM kinase activity by giving it these little spikes. And then if you lose that, then it just stops being active and drives it towards differentiation.
And does CAM kinase then intersect with the network of the hairball of different signaling pathways?
Yes,
Yes.
and I don't remember how, but like
Ah.
if you want to, you can tie calcium signaling to every pathway.
Yeah.
So I mean, and I think we even did that in the paper. I just don't remember off the top of my head. Yeah.
Great talk. I was wondering if you looked at whether the cells that participate in a calcium wave are clonally related. I wonder if it's something like the follicle cell syncytiums that you have in lymph gland.
Yes, that's a good question, because I mean, this is something we've obviously thought about and try to make reviewers happy about. Is I mean, because why is it only traveling through these few cells? Why? Because the calcium the gap junctions are throughout. They can connect other cells. So why is it just traveling through the 5 cells that we see? So they could be clonally related, and this is something we could look at. That's also, I think, a wonderful suggestion. Yeah.
Okay.
Good. This was wonderful. Thank you so much, guys. Yes.
Okay. Thank you. Okay, so our next speaker will be Prash Rangan from Mount Sinai.
Good morning, everybody. First, I'd like to thank the organizers for inviting me to this fantastic meeting. I've had a lot of fun so far, and I'm super excited to be here talking about my lab research program, where we've discovered a new programmatic transition during germline development. First, I would like to start by thanking the people who did the work. Most of the work I'm going to show you today was done by 2 graduate students, Kaini and Noor, who are in the audience here. as well as 2 previous graduate students, Alicia McCarthy and Patrick Blatt. This work would not have been possible without the help of collaborators listed there, but one person who I want to highlight is Michael Buzak, who shared an unpublished reagent with us, which made all the work today, what I'm going to tell you today, possible. And last but not the least, I want to thank the funding agencies for keeping the lights on. My lab works on germ cells. These are the cells that give rise to gametes, egg, and sperm. We work on germ cells because these, upon fertilization, give rise to all your somatic cells in the body, as well as new germline. This new germline continues its march through generations, giving rise to a new organism in every generation. Okay, so therefore, germline or germ cells are both immortal as well as pluripotent. Among the germ cells, we are specifically interested in looking at the oocyte. Why is that? While most organisms require sperm and egg to launch the next generation, some organisms have dispensed with sperm altogether, right? And can do this by a process called parthenogenesis. Not only that, you can take an oocyte and use it for somatic nuclear transfer and make all the somatic cells in your body, right? So understanding the biology of the oocyte can give you an insight into molecular basis of pluripotency and, if you will, immortality. And this is the research focus of my lab's research program. Oocytes arise from germ cells, as I told you, and germ cells are among the first cells to form in a fertilized zygote. These germ cells, once they form, will migrate to the gonad. In the gonad, they have 2 options. One, they can directly differentiate and enter meiosis. This happens in women. Or they can become germline stem cells, which self-renew and differentiate to give rise to oocytes, as in the female fruit fly. So since I'm at a fly meeting and this is the model we are using, I'll give you a brief primer about the Drosophila oogenesis. Each female Drosophila has 2 ovaries. Each ovary is made up of individual units called ovarioles, shown there. Each ovariole is an assembly line of egg production, okay? This assembly, egg production starts at the anterior-most end of the ovariole in a structure called the germarium. This schematic of the germarium is shown on the top and the pictograph is shown below. The germarium broadly consists of 2 kinds of tissues. The somatic tissue, which provides structural support and signaling pathways to maintain the germline. Egg production in the germline starts at the anterior-most end by 2 germline stem cells that are present. Okay, these germline stem cells are marked by these round structures called spectrosomes. Once these germline stem cells self-renew and divide to give rise to a daughter, they express a differentiation factor called bag of marbles, or BAM. Once you express BAM, they divide 4 times incompletely to give rise to 2, 4, 8, and 16 cell cysts. Okay, these cysts are marked by these extended structures called fusome that are shown here. So among the 16 cells in the cyst, 15 cells become nerve cells. These are support cells that make mRNAs and proteins that give rise, that give all these products to the growing oocyte. Also, several cells in the cyst will initiate meiosis, but only the oocyte will get committed to meiosis, okay? So together, they will form an oocyte and then bud off from the germarium, forming the egg chamber. So the purpose of for the purpose of today's talk, I want you to know 2 things. One is that the germline stem cells and the immediate progeny are in the mitotic phase of cell cycle, whereas the cysts, and as they differentiate, are in the meiotic phase of the cell cycle, okay? So once you form the oocyte, as I told you, they form the egg chamber. Once you form the egg chamber, what happens is as the nerve cells provide more mRNAs and proteins, the egg chamber grow in size, eventually giving rise to a mature egg, okay? And then the nerve cells die back, leaving just the egg alone. My lab's research program is interested in understanding how do you go from the germline stem cell all the way to the mature egg, okay? For this, we broadly divided this whole process into 3 transitions. One, the stemness program, where the germline stem cells divide and differentiate to give rise to the progeny. And then once the progeny is made, these progeny will then enter meiosis and commit to meiosis to make an oocyte. Once the oocyte is then made, the nerve cells, as they increase amount of mRNA production and proteins, they will make these maternal products, or the maternal contribution. So we published on all three aspects of this, so please check out these papers. But for purpose of today's talk, again, we're going to focus on one specific transition, that is meiotic entry and commitment, or the meiosis promoting program. What do I mean by that? So what I mean by meiosis promoting program is the idea of meiosis 1. This is a wild program if you think about it, right? The cells undergoes, there's homologous recombination by pairing of chromosomes, and then you can have DNA double-strand breaks, and then you cross over pieces of DNA utilizing the specialized machinery called synaptonemal complex. If this wasn't crazy enough, then it also undergoes another crazy process called reductional cell division. Okay, this reductional cell division is mediated by proteins such as RBFOX, which helps shift from the mitotic phase into the meiotic phase of the cell cycle. Okay. So if this were to happen in any other cell type, that cell will end up having an SOS signal and resulting in cell death. But the germ cells do it reliably and give rise to the next generation over and over again. Okay, so the question then becomes, how do you enter this meiosis 1 program and how do you exit this meiosis 1 program? The entry into meiosis 1 program, we are really interested because this is conserved in all sexually reproducing organisms, right? And for something as fundamental and conserved as this, still don't know how this really works. And the next question we wanted to ask is, how does exit work? And when I say exit, I don't mean exit from meiosis, because oocytes are meiotically arrested. We are talking about how do you exit meiosis 1 initiating factors. For example, if you think about it, if you make a lot of proteins for synaptonemal complex and form it, do you even need more of it, right? Do you need keep making all these specialized proteins, or are they shut down? We have no idea how this works. So we published on the entry, and I'm going to summarize this in a few slides. So what we found, utilizing bioinformatics as well as genetic screens in Drosophila, that entry into meiosis is regulated at the level of both transcription as well as translation. And so what do I mean by that? What we find, and what I found it really shocking, is that you make these meiosis 1 promoting mRNAs in the mitotic stages of the cell cycle. But these mRNAs are not translated there. It's a little suicidal if you think about it, right? And these mRNAs are then undergoes translation only in the meiotic stages of the cell cycle, allowing for entry into meiosis to happen. And this is in part mediated by a chromatin reader called a chromatin reader of H3K36 marks. So in specific, what we found, and what I must confess is the mechanism I'm going to show you today is an inferred mechanism, okay? We found that set 2, which deposits H3K36 marks, is required for meiotic entry. This H3K36 marks recruits a reader of H3K36 marks called male specific lethal 3, or MSL3, okay? This MSL3, what it does then is recruits another complex called the ATAC complex, which then deposits H4K16 as stylation mark, right? You may ask, because I asked this question, why all this drama? So we think that this drama is to specifically deposit this H4K16 marks on a specific subset of genes. The property of the H4K16 marks is that it unpacks nucleosomes, allowing for transcription to happen. So when this pathway is active, it has two kinds of effects. First is that it promotes transcription of the synaptonemal complex mRNAs such as SUN, ARD, and CONA. Okay. Then it also promotes transcription of a ribosomal protein, RPS19B. This ribosomal protein gets incorporated into the ribosomes and promotes translation of a meiotic cell cycle factor, RBFOX, which I told you about. Okay. So together they promote entry into meiosis so that you can start life, initiate life. So from this, what I'm trying to say is that we have data that shows that meiosis promoting program utilizes special chromatin machinery, and this chromatin machinery both transcriptionally and translationally coordinates entry into meiosis. The next question we had is what happens to these genes? Are they silenced after meiosis entry is initiated? And if so, how? And the reason why we're asking this, an active process would suggest a programmatic transition. Okay. And so before I tell you, I would like to flash this slide again and tell you that I'm going to describe all my results in the eyes of two proteins, if you will. One, RPS19B, which promotes meiotic cell cycle. Another one is sisters unbound, or SUN, which is required for recombination. This is in part because reagents were available. So when we looked at RPS19B expression, what we found was RPS19B is expressed in the mitotic stages as well as the early meiotic stages, and its expression was attenuated as soon as the oocyte was specified. Okay, this is the beginning of the maternal program. And if you're not convinced by this, you can take a step back and look at the whole ovariole. You can see that RPS19B is expressed at the very early parts of the germarium and then shut off at the later stages. So the question here is that what causes silencing of RPS19B in the later stages of oogenesis? So Kahini Sarkar, who is in the audience, carried out a screen, and she found, and I should say this is a preprint you can check out already, found that regulated heterochromatin silences these genes at the as soon as the oocyte is specified. And the crazy part is that we found that regulated heterochromatin is in a feedback loop with the nuclear pore complex to maintain the silencing of these genes. And it's absolutely essential to silence these meiosis 1 promoting genes to initiate the next program, which is the maternal program. Okay, so in a screen, what Kahini found was that ZDB1 was involved. ZDB1 is a histone K9 methyl 3 depositing enzyme. Okay, when such marks are deposited, you can take euchromatin and make it heterochromatin. Okay, so when you lose ZDB1, you lose H3K9 methyl 3 marks and cause gene upregulation. And beautiful work published from Chule Jesus Riggs' lab has already shown that ZDB1 is mostly cytoplasmic in the mitotic stages and becomes nuclear as meiosis progresses, showing that this protein is there at the right time and right place to do the job. And what Kahini found was that loss of ZDB1 results in upregulation of RPS19B in all the late stages of oogenesis. That's shown over here, right? So then you may ask, okay, this is great. Is this direct or indirect process? Okay. So the question now becomes, if it's a direct process, then we should observe these H3K9 marks in and around genes that regulate entry into meiosis 1, right? So again, to test this hypothesis, we carried out cut and run experiments utilizing H3K9 methyl 3 marks. So these cut and run experiments immediately show that in control, which is in the later stages of oogenesis, you have beautiful H3K9 methyl 3 marks on loci such as RPS19B as well as SUN. Okay. And this levels of H3K9 is attenuated upon loss of ZDB1, showing that ZDB1 indeed directly regulates a cohort of meiosis promoting genes. Okay, so from here we concluded that regulated heterochromatin, and we say regulated because there is a regulated translocation of ZDB1, promotes direct silencing of meiosis 1 promoting genes to cause their silencing. Right. So this could have been done. This is all great. And then what Kahini did was she told me something really strange and bizarre. And I was like, what is going on here? Okay. So the next part of the story may sound tangential at first, but it's going to make sense later on. So what Kahini observed was that in the down-regulated targets of ZDB1, she found 18 of 30 members of nuclear pore complex to be not expressed properly. This is kind of wild if you think about it, right? So nuclear pores, as you know, promote transport of macromolecules in and outside the nucleus. But this excited for like many reasons, including because there were recently published papers in yeast that showed the nuclear pore complex were part of the heterochromatin proteome. And not only that, that these nuclear pore components help anchor and maintain heterochromatin. Okay, so this led us to ask two questions. First question was, is it possible that when you lose heterochromatin, you're actually losing nuclear pore complex members or nuclear pore complex? And the second question is, does in Drosophila, like in yeast, when you lose nuclear pore complex, do you lose heterochromatin formation? Okay, so to answer the first question, we stained for nuclear pore complex in both control and ZDB1 depleted ovaries. So the control is shown over here, and you can see beautiful nuclear pore complex formed around the nurse cell nuclei. When you lose ZDB1, you lose this beautiful staining around the nurse cell nuclei and form these punctae, which I have no idea what they are, okay? But this, I don't know about you, this is rather wild for me, right? If you lose heterochromatin, now you're not forming nuclear pore complex. So now the next question became, is it possible that this nuclear pore complex also maintains heterochromatin like in yeast in Drosophila? To test that hypothesis, what we then did was again carried out cut and run experiments and for H3K9 methyl 3. But this time we did it upon depletion of a nuclear pore complex member, NUP154. Okay. NUP154 is a germline enriched member, and a phenotype has been previously described before. And again, as in loss of ZDB1, we find that there's beautiful enrichment of H3K9 methyl 3 on loci such as RPS19B and SUN. And when you lose when you lose NUP154, you lose this enrichment. So this led us to a model. The model is that now we have regulated heterochromatin, which does two things. First, it silences meiosis-1 promoting genes that allow for transition from GSC to an oocyte. The other thing it does is increases the level of nuclear pore complex, which then maintains heterochromatin itself. So obviously you may ask, where you don't have nuclear pore complex and when you don't have regulated heterochromatin formation, you get this dysregulation of meiosis-1 promoting genes, okay? So is there a bad consequence for when these meiosis promoting genes are present in the maternal stages of oogenesis? So essentially what we are asking is, now you have RPS19B expressed in the maternal stages, is there a problem with these stages, okay? So for this, we stained for a protein called egalitarian. Egalitarian is an RNA binding protein that actually binds to these maternal mRNAs and transports them into the oocyte. So when you look at the control, egalitarian, and I should say it's a really good proxy for the maternal program. When you look at the control, you can see beautiful egalitarian accumulate in the future oocyte. But when you lose ZDB1 as well as NUP154, what happens is there is initial oocyte specification, but this is not maintained and it's completely ablated at the later stages, okay? So what I want to do is now summarize this part of the results, saying that now we have a specialized transcriptional machinery to upregulate the meiosis-1 promoting program, which coordinates transcription and translation. And now we found that there is a special feedback mechanism between heterochromatin and nuclear pore complexes that silences meiosis-1 promoting program. And this silence is absolutely essential for kickstarting new program or the maternal program, okay? So obviously you may ask, or at least I asked, is what controls this transition at this particular stage, right? How does RPS19B decrease at the cis stages and allow upon oocyte specification? And I should add, this is unpublished work that was carried out by Noor Kut, and the answer to this was shocking for me. We found that ribosome biogenesis and translation is absolutely essential to mediate this transition. We found that this happens in part through translation, again, of the nuclear pore complex members, okay? So what did we do and how did we find this? Again, a screen in Drosophila, Noor found that there are several proteins of the ribosome biogenesis pathway like bistin and ZFRP8, as well as the members of the TARC1 pathway, such as Raptor and Meos, are involved. The clue that this was a transition came from looking at TARC1 activity, okay? So as you know, TARC1 is a kinase that phosphorylates several enzymes. It has many effects, including increasing translation, as well as increasing ribosome biogenesis. And one of the targets is our ribosomal protein S6, which can be phosphorylated. And if you look at this activity, you can see again in the cis stages, its activity is really high. And beautiful work from Mary Lily's lab has shown that this increase in TARC1 activity is required for meiotic entry itself. And to our surprise, we found that loss of TARC1 results in upregulation of RPS19B in the later stages. Again, so if we lose Gator2 complex, which is a positive regulator of TARC1 activity, or members of the TARC1 complex itself, we get this expression of RPS19B in the later stages of oogenesis compared to the control. This is not just limited to the TARC1 complex. We can lose members of the ribosome biogenesis pathways, such as bistin and ZFRP8, and we get the same thing, okay? So this suggested to us that there is something special about translation that's happening at this particular stage that allows for silencing of RPS19B. So what are the things that may be controlled at the level of translation, okay? To determine what proteins or what mRNAs are being controlled the level of translation, we carried out polysome-seq analysis. So essentially what this is you purify polysomes. And polysomes, as you know, have multiple ribosomes present on the mRNA. Okay, so the idea here is that if you do a control and if you have conditions where ribosome levels are lower or TARC1 activity is lower, that you may see a differentially translated group of mRNAs. And so indeed, we purified polysomes and we sequenced them, compared how much mRNA is inside these cells. And we found that several mRNAs were downregulated at the level of translation, which is known as translation efficiency. Again, when we looked at what these proteins were, we were kind of stunned. These proteins were, these mRNAs were members of the nucleopore complex again. These nucleopore complex members were either downregulated at the level of mRNA and polysomes, or polysomes alone. So this led us to, again, have this hypothesis where now we are saying that levels of ribosomes control levels of nucleopore complex. This is, again, wild for me if you think about it. I don't know, it may be like, when I heard this, I was like, what? Right, so question is, if you reduce ribosomes, do nucleopores form properly? And this is exactly the question Noor asked. And again, when you use a control, we found beautiful nucleopore complex around the nurse cell boundary. Whereas when we reduce ZFRP8 as well as MIO, which is part of the TARC1 pathway, we lose this expression of nucleopore complex. And what I've already told you is that this nucleopore complex is absolutely essential to maintain heterochromatin or to cause silencing of these meiosis I proteins, right? Meiosis I genes. So then the prediction should be that if you lose ribosome levels or TARC1 activity, you should also lose heterochromatin. To test this, we again carried out cut and run, but we now did cut and run against H3K9 methyl 3 in ribosome biogenesis mutants as well as TARC1 activity mutants, such as MIO as well as ZFRP8. And I'm showing you results for the RPS19B locus. Again, you see beautiful enrichment of RPS19B, or heterochromatic enrichment of RPS19B locus, but this enrichment is lost upon loss of ZFRP8 as well as MIO. So these results are exciting to us for two reasons. One, actually many reasons, but I'm going to tell you two reasons. First is that meiotic entry is controlled by TARC1 from yeast, at least till Drosophila, right? What we are finding is the very same pathway that promotes entry into meiosis also promotes translation of the silencing machinery that then stops it, okay? And also the other idea we have is that now we're seeing changes to the level of translation can affect the chromatin state of a cell, right? So if you have transient changes in levels of translation, this can be caused by metabolic changes and so on and so forth. This can be encoded at the level of chromatin, which is kind of wild for us if you think again, right? And then that leads to a conclusion where we now have a meiosis I promoting program that is activated by a transcription machinery. And we need translation to make very many activators of this process. And the same upregulation of translation at this particular stage also translate the repressive machinery to silence them. Once you make this repressive machinery, then you make stable silencing of these genes throughout rest of oogenesis. And this silencing is required to absolutely initiate the maternal program. And so now I've told you about entry into meiosis I and exit from meiosis I at the transcriptional level. So last question we had is, okay, now you stop making these mRNAs at the transcriptional level. So what happens to the mRNAs that are already there that are making proteins? And I will not give you more data. You can be happy about this. I'm going to summarize the slide in one slide. So what we found was that at that particular stage, again, in the cis stages, these mRNAs undergo regulated RNA degradation. And this is mediated by components of the no-go decay pathway. Okay, you can read about this, it's already published. So this then leads to a bigger picture that's evolving, right? The bigger picture is that there is a programmatic transition that's occurring at the onset of meiosis that helps to both activate and silence these genes as you transition from a germ cell fate into an oocyte fate. And this transition we have termed a germ cell to maternal transition, and I'll tell you why. So if you zoom out from this transition, you already may know that the maternal program initiates life, right? This maternal program is degraded during the maternal to zygotic transition that's been worked on extensively by Howard, who is in the audience over here. Once you degrade this maternal program, you initiate new life and new identity via zygotic transcription, which Melissa will talk about next. So what we are finding at the other side of this transition, that you have meiosis 1 mRNAs that are silenced and degraded in the very same way. But what I didn't tell you, and we are finding, is that several of the targets also involve germ cell genes that are present at earlier stages, as well as germline stem cell genes that are present at earlier stages. So that's why we are terming this transition maternal to germ cell to maternal transition. So if there is one thing that I want you to take home from today, is that when you want a fresh start in life, you want to get rid of old baggage and then start afresh. And this happens at both ends of your oocyte program. And so I strongly do believe that there is something to be learned about reprogramming cells from here and for regenerative therapies. So with that, I would like to stop and take questions. And also I am actively looking for postdocs. I'm sure if there are questions, there's going to be several which we don't know the answer to. So if this excites you, reach out to me.
It was a great talk, Prash. Thanks.
Thank you.
Do you see or have you looked at in the SETDB1 mutants when you see decrease in nuclear pore complex factors? Does that affect do you see differential mRNA export out of the nucleus, and is that a mechanism of the regulation?
Right. This is super, super exciting. So first of all, thank you for bringing that up. This is also crazy for me. These cells don't have nuclear pore complex. They are fine till the end up dying in around stage 7, right? How do these cells even function without proper nuclear pore complex? We have no idea. The second question is the export and import affected? At least for the members we've checked, it's not, because RPS19B mRNA is going out to the cytoplasm and is being translated there. And sometimes you can see RPS19B going back into the nucleus where, and becoming part of the nucleolus, right, where ribosome biogenesis takes place. So I have no idea how this is happening. And so, yeah, but the transport seems to be okay, at least for the things we've tested.
Do you know how SETDB1 and canine methyl is targeted to the genes that have to get shut off?
Right.
When you turn off meiosis?
Right. This is a great question. So how do you specifically target heterochromatin to meiosis 1 promoting genes? So in Drosophila, in mammalian cells, you have crab proteins that actually direct deposition of heterochromatin at that specific genes. And Drosophila don't have crab. So we screened and we found members, other transcription factors that are recruiting SETDB1. We think is recruiting SETDB1 to those specific loci. And we don't have the full mechanism as yet, but.
That leads into my question perfectly. So small ovary might be a crab-like, have a crab-like function. Have you looked at that or run into it?
No, but it's on a short list.
Okay.
Thank you. Yeah.
You over here, you talk about getting rid of all transcripts. Do you see any evidence of an effort to get rid of all proteins? Because you got to get rid of those also.
If there's any postdocs looking for this question, come reach out to me, because this is an important question, right? And we are really so, and that's an excellent question, because at that very particular stage, my colleague and my postdoc friend, Thomas Hurd, finds that old mitochondria will, I don't know, fission, whatever he calls it, and then they're turned over, right? So the mitochondria being turned over at that stage, and we are finding this whole program that's happening at this stage, and what happens to old proteins. Then this is something we are really excited by, and we have it'd be great to study it. All right.
All right.
Thank you.
All right. Well, it is my pleasure to introduce Melissa Harrison. Take it away.
Thanks. All right. I first want to thank the organizers, not only for inviting me, but for this wonderful meeting that has really re-energized me. Obviously, all of you for being here, and in general, the Drosophila community for being just a really fantastic and welcoming place to do fun science. So with that, what my lab is really interested in addressing is how do you take these two highly specialized germ cells, the sperm and the egg, put them together and get an entirely new organism. And so if we think about what has to happen after fertilization, that single-celled zygote goes on to divide and differentiate into all the cells of the adult organism. But if we look at, let's say, a neuron in the brain versus a muscle cell in the heart, those cells are phenotypically distinct and clearly built for the job they're meant to do. But those differences in phenotype can't be due to differences in genotype because they all arose from that same single cell and share a DNA genome. So what we're interested in then is how is that DNA genome differentially interpreted over the course of development to give rise to all of these different cell types, and how when misinterpreted, it can give rise to disease. So if we think about this, we know for thousands of organisms the sequence of that DNA genome. And we know about hundreds of proteins, transcription factors that can bind to that genome in a sequence-specific manner. And so in some ways, it seems like it could be a computational problem, but biology gets in the way. So if we look here at 8 transcription factors in the early Drosophila embryo, and we look at the percentage of sites that they bind in vivo based on their in vitro prediction of binding, we can see that it's only about 5 to 15% of those sites that are occupied, which means 85 to 95% of the time that sequence prediction is wrong. And that's because the DNA is not sitting naked in the eukaryotic nucleus like it is in a test tube. It's wrapped around histone octamers to form nucleosomes, and these nucleosomes are further compacted into secondary and tertiary structures to fit that giant genome in that tiny nucleus. So if we take a step back and we now look at these proteins, these transcription factors, and think about the fact that they can bind to that sequence when it's open and accessible. But we know that when that sequence is wrapped around a histones to form nucleosomes, that can be a barrier to transcription factor binding. So it's really that more limited sequence space of open and accessible chromatin that transcription factors are interpreting. So a more direct question that we're interested in addressing is how is chromatin accessibility modified to influence transcription factor binding? And so a model was put forth based on a canonical factor called FOXA by Ken Zarrett and others, that there were specialized transcription factors called pioneer transcription factors. And these factors can bind to nucleosomal DNA. They can then establish these accessible chromatin domains, and they can now facilitate the binding of additional transcription factors and the activation of gene expression. And we know that proteins with these characteristics are fundamental in developmental transitions. So a good example is a cocktail of transcription factors that can reprogram specified cell types to induce pluripotent cells, include OCT4, SOX2, and NANOG, which all have these pioneer factor characteristics. And it's not just in culture, but we know that this happens in reprogramming in vivo, during differentiation, and in transdifferentiation. So my lab is really interested in understanding what are the essential features of these pioneer-like factors, and how do they drive these dramatic cellular transitions? And to do this, we study conserved developmental transitions. And one of the ones that we're focused on is this maternal to zygotic transition. So immediately following fertilization, development is controlled by maternally deposited mRNAs that we just heard about from Prash. So it's a good job, organizers, for the synergy. And these maternal RNAs control the initial stages of development because the zygotic genome is transcriptionally quiescent. And it's not until hours or days later that the zygotic genome becomes transcriptionally active. And there's a coordination of the activation of the zygotic genome with the degradation of those maternal mRNAs. And while this transcriptome of the embryo is being reprogrammed, so is the developmental state, as it moves from those two highly specialized germ cells to the totipotent cells of the early embryo that can then give rise to all of the adult cells. And so if we think about this transition, and we know that in all organisms studied to date, pioneer factors drive the activation of the zygotic genome. So if we look at this transition from these specified cell types to these pluripotent cells during this maternal to zygotic transitions, there are parallels to that reprogramming that I told you about in culture. And this parallel was made explicit by the demonstration that orthologs of OCT4, SOX2, and NANOG activate the zygotic genome in zebrafish. And the first activator of the zygotic genome that was identified was Zelda, identified by Chris Ruslow's lab in Drosophila. And so what we're interested in using this transition to understand What I'll tell you about today is what pioneer factors reprogram the early embryo. Is constitutive activity of these factors important in this in driving this developmental change? And then also, is the Zelda-mediated reprogramming that we see in the embryo limited to this developmental time point, or does it have functions outside of the early embryo? So to start, I want to bring everyone up to speed. I know not everyone's as excited about the early embryo as I am. So what is happening in the fly embryo at this time point? So it's characterized by a series of 13 synchronous nuclear divisions. These are occurring every 10 minutes. So there's only time for a mitotic division cycle and then DNA synthesis. And as I said, this is being controlled by maternally deposited, primarily mRNAs. And then the zygotic genome initiates transcriptional activation at approximately cycles 8 to 10 with an early minor wave of gene expression. And this is gradually activated with widespread zygotic genome activation occurring at nuclear cycle 14. At this time point, this cycle slows, the division cycle slows, a gap phase is added, and this nuclei are cellularized. So Zelda is maternally deposited as an mRNA and translationally upregulated at nuclear cycles 8 to 10. And I hope you saw the poster from Liz and Zoe in my lab about this regulation. And so as a postdoc, I used ChIP-seq to identify where Zelda was binding at this early time point. And those data showed it bound thousands of regions of the genome, but we already identified something distinctive about Zelda. As opposed to those other transcription factors that I showed that only occupy about 5 to 15% of the sites they're predicted to bind, Zelda occupied 64% of the sites it was predicted to bind, which is approximately 10 times as much as those other transcription factors. Furthermore, when we assayed Zelda binding at multiple discrete time points spanning the maternal to zygotic transition, we saw that once established, Zelda binding was largely retained. And so these data and others suggested that Zelda had key characteristics of a pioneer factor, right? We showed that the binding was dependent on DNA sequence, suggesting it was agnostic to the presence of a nucleosome. And then work from Chris Rushlow's lab, Alex Stark's lab, and Steve Small's lab showed that Zelda could facilitate the binding of additional transcription factors. So what we really wanted to set out to ask was whether Zelda was required to establish or maintain accessible chromatin, a key feature of pioneer factors. So we made germline clones to deplete maternal Zelda and looked at the top accessible regions in the early embryo. And since we were interested in Zelda's role in driving these accessible cis-regulatory regions, we asked if they were bound by Zelda by overlaying our ChIP-seq data. And then we could ask whether they depended on Zelda for accessibility by looking at accessibility both in wild-type control embryos and those maternally depleted embryos. So we identified 400 regions that depended on Zelda for accessibility. And these are exactly where we saw Zelda behaving as a pioneer-like factor. We could show that not only did these regions depend on Zelda for accessibility, but also other transcription factors needed to bound to these regions needed Zelda-mediated accessibility to bind and to activate gene expression. But what was surprising was we identified 1,500 regions that, while bound by Zelda, remained accessible in its absence. And to us, this suggested the possibility that there were additional pioneer-like factors governing accessibility. So we compared we looked at a lot of comparisons between these two Zelda-bound regions to identify what might be distinctive about the two classes. And what we identified in these regions that did not depend on Zelda for accessibility was they were enriched for the GA dinucleotide. And so there are a number of proteins that bind GA dinucleotides. And we were particularly excited by GAGA factor, or GAF. And that's because beautiful work from a number of labs had shown in tissue culture primarily that GAF had roles in governing chromatin accessibility. It interacts with proteins that are known to move nucleosomes about, and it has a role in three-dimensional chromatin structure and RNA polymerase pausing. So we wanted to ask, is GAGA factor an additional pioneer factor that governs accessibility during this reprogramming event? But we ran up to a roadblock, which was that we couldn't use the same trick to deplete GAF that we had for Zelda, because getting rid of GAF in the germline did not result in embryos. Furthermore, it had been reported that GAF RNAi didn't work in the early embryo. So instead, what we did was we added a GFP tag to the N terminus of Zelda using Cas9 mediated engineering. And then we could build on the system that Marcus Alfelter's lab developed, in which they fused a nanobody that recognizes GFP to an F-box protein. And when this F-box nanobody fusion is expressed, it can recruit GFP tag proteins to the ubiquitin machinery and lead to their degradation. So when we express this protein in the early embryo, what we could identify here, we have nuclei. You can see the nuclei marked with a fluorescent histone, and then we could see our GFP tag GAF. But when we co-express this degrad fusion, while we can see the embryo develops, there is no, or at least not detectable GAF. So now we could use this system to ask what happens to these embryos that don't have GAF. And we found that 100% of them died, showing that GAF is essential for this maternal to zygotic transition or embryo development. We could also use these degrad embryos to specifically ask the question we wanted to understand. Is GAF important in determining chromatin accessibility during this early reprogramming event? And so we performed a technique called attack-seq. And here I'm just showing a single gene in which we see a genome, and we can see these peaks that correspond to chromatin accessibility. And then we could identify regions like this that lost chromatin accessibility specifically when GAF was degraded. And we could overlay these with our ChIP-seq for GAF and show that there were regions that depended on GAF and were bound by GAF. And indeed, when we look genome-wide, I'm showing you here a volcano plot where we have the log 2-fold change on the x-axis and the adjusted p-value on the y-axis. So anything to the left is a loss of accessibility and anything to the right is a gain of accessibility. We can see that hundreds of GAF-bound regions lose accessibility when GAF is depleted, showing that GAF is indeed important in determining chromatin accessibility in the early embryo. We also performed RNA-seq and could show that similarly, like here at this example of UBX, that GAF is also essential for activating the zygotic genome. And so we wanted to start to understand, how do these two now pioneer factors, ZELDA and GAF, potentially work together? And we did a bunch of work, but one of the things that I'll show you here, because we're interested in chromatin accessibility, is that we could take advantage of beautiful data from Shelby Blythe, in which he did single embryo attack-seq at very precise time points spanning this transition. And we could look at when this accessibility was established. And so there's some regions that are already accessible at nuclear cycles 11 and 12. And then there are other regions that become progressively accessible, like these, at nuclear cycles 13 and 14, when that widespread zygotic genome is being activated. And what we could show is that, like this individual area, that regions that bound by GAF and depend on GAF for accessibility, they become accessible later. They're not accessible early. By contrast, regions that depend on ZELDA for accessibility are already accessible as early as nuclear cycle 11. And so together, what these data showed us was that ZELDA appears to be primarily the most important. It's important throughout, and I'll talk about that next, but the most important for activating this early wave of gene expression by mediating chromatin accessibility. By contrast, GAGA factor comes on slightly later to coordinate and to further refine the accessible regions in the early embryo. And then Erica Larson's lab has shown that CLAMP, another GA dinucleotide binding factor, is also important for this. And then OPA has been shown to be important in gastrulation, a pioneer factor, by both Shelby Blythe and Angela Stathopoulos. So what's really nice about this is now we have this really beautiful in vivo event in which we can have multiple pioneer factors that are coordinating to reprogram from the specified cell type to the early embryo. And so there's now this also sort of satisfying similarity to the other reprogramming events, whether it's in culture or the activation of the zygotic genome in other species that involve multiple pioneer factors. And we can see that Drosophila are not distinct by having a single. There are, again, multiple factors that have to work together for this transition. So now we could use this to ask more general questions about pioneer factors. And so we wanted, knowing that ZELDA had these pioneering characteristics, we wanted to ask whether continued pioneering activity was required to maintain these gene expression profiles once established, or whether by contrast, once the pioneer factor had established the accessible chromatin, these downstream transcription factors had come in, whether the pioneer factor was now not necessary. And so if we think about that in terms of ZELDA, what we wanted to know was whether ZELDA activity was required throughout zygotic genome activation, or instead whether Zelda activity was only required during those early stages to activate that early wave of gene expression, which includes many of these downstream transcription factors, and that once they had bound and established this, could they now maintain and activate widespread zygotic genome activation? So to address this question, we had to get rid of Zelda during this time point. But this is all occurring in 1 hour of development and over 4 nuclear divisions. So this was going to be way too fast for us to use proteasomal mediated decay. So we thought that an optogenetic system would give us the temporal resolution we needed. So what we did was we added a blue light responsive CRY2 tag to the N terminus of Zelda using Cas9 gene editing. And normally Cry2 is part of a 2-component system, but as we were developing our system, Timothy Saunders' lab showed that blue light alone could inactivate a Cry2 tag transcription factor. So we wanted to test this for our Zelda tag. And so we knew that the Cry2 tag alone didn't inhibit the essential functions of Zelda because the flies are homozygous, viable, and fertile. And we knew when we treated embryos, wild type embryos with blue light, they can, I'm showing you here, enter nuclear cycle 14, and then an hour later have this stereotypical gastrulation. But when we treated our Cry2 tag Zelda embryos with blue light, they could enter nuclear cycle 14 with some defects, but there was catastrophe and they failed to gastrulate. And this phenocopies the Zelda maternal loss of function. And we could also then wanted to test whether this was specifically inhibiting transcriptional activation. And so we took single embryos, started shining blue light on them as they entered nuclear cycle 10, kept blue light on them throughout the early and major waves of zygotic genome activation, and harvested single embryos 30 minutes into nuclear cycle 14. And we did control embryos that were exposed to blue light as well as Cry2 in the dark. And we did single embryo RNA-seq. And so I'm showing you here 3 well-known Zelda targets, Bro, Foots, and Halo. And we can see they are robustly expressed either in the Cry2 embryos without exposure to blue light or in wild type embryos exposed to blue light. But there's very little to no expression in the presence of blue light. However, this other gene is, there is RNA. And so we can look again genome-wide. And what we identified were hundreds of downregulated genes, and these were highly enriched for those that had Zelda binding sites nearby. So this really confirmed to us that this method was inactivating Zelda. And I hope that you got to see Audrey's talk from the lab in which she's used this technique to inactivate CBP. And now with this tool in hand, we could ask the question we specifically went out to address. So we now shown blue light on the embryos either as they enter nuclear cycle 13 or nuclear cycle 14, and again, harvested them 30 minutes into nuclear cycle 14. I'm not going to show you the data, but I will tell you that the conclusion was that indeed continued Zelda activity was required for this widespread zygotic genome activation. So even when we shone blue light only during this short window, we did not see robust activation of those zygotic genes. We could also use this tool to ask the opposite question. What happens if we just shone blue light during this early wave, then allow the embryos to develop in the absence of blue light until 30 minutes into nuclear cycle 14? Could this pioneer factor now, even when not active earlier, could it drive gene expression during the major wave? And indeed, we showed that it could. And so what this really told us was that Zelda activity, like Zelda binding, was required throughout this transition. And this includes at least four mitotic division cycles. So how is this Zelda binding and activity maintained? And one of the things that we thought is that it had been shown that for a subset of pioneer factors, they can be retained on the mitotic chromosomes, while many transcription factors are evicted from the highly compacted mitotic chromosomes. So we thought maybe Zelda could remain on the mitotic chromosomes, which could explain how its activity and binding were maintained. So we have GFP tagged Zelda, endogenous Zelda, and here I'm showing the chromatin with a fluorescent histone, and we can watch what is happening through these beautiful synchronous nuclear divisions. And I hope what you can appreciate from this movie is that it is not mitotically retained. It's actually eliminated from the mitotic chromosomes, and then robustly Zelda's binding has to be rapidly and reproducibly reestablished during DNA synthesis phase. And so again, we turned to the definition of a pioneer factor and thought about the properties that might enable Zelda to have this. And that is that, again, I've told you that pioneer factors can bind to DNA even in the context of a nucleosome. So we took DNA that corresponded to a Zelda binding site in the embryo. And we could do electromobility shift assays with full-length Zelda. And here I'm showing you the naked DNA, that's about 160 base pairs. And when we add the full-length Zelda protein, we can see this shift showing binding. And we collaborated with Peter Lewis's lab to make nucleosomes with this DNA, and again, could do these shifts. And we could see that Zelda could bind to the nucleosomal DNA. Furthermore, we could show that this binding was sequence, at least partially sequence specific, and that this shifted band included histones. And so what this told us was that Zelda was able to bind to its DNA even when it's wrapped around a nucleosome. And so together, what these data led us to understand about this pioneer factor was that this activity was required throughout these transitions, and that despite the fact that there's all these synthesis and mitotic phases, that we think what is happening is that Zelda is binding and establishing or maintaining these nucleosome depleted regions. But as the replication fork passes, we know that nucleosomes are reestablished. Work from Shelby Blythe and from Steve Hennicoff's lab has shown this. And so we think that it's this pioneer factor's ability to recognizing its binding site, even in the context of a nucleosome, that allows it to really rapidly rebind and reestablish these nucleosome depleted regions. And so using these tools, we've been able to show that continued activity of this pioneer factor is important for defining these cis-regulatory regions and activating gene expression. Now, I've just told you that the chromatin environment in the early embryo is unique, right? There are these rapid replication cycles, but it's not just that. There's actually a distinct linker histone that's replaced by the canonical H1 at nuclear cycle 14. And there's also very limited histone modifications. So now we were interested in, is this Zelda-mediated reprogramming that we identify in the embryo unique to this developmental time point, or can Zelda reprogram outside? And we already had some hints from the original work by Chris Rushlow, in which she showed that not only is maternal Zelda required for viability, but zygotically expressed Zelda is also required. So we wanted to identify another tissue type in which to study Zelda. And we collaborated with Cheng-Yu Lee's lab and looked in the type 2 neuroblasts of the larval brain. And these are neural stem cells that divide asymmetrically to self-renew and also to produce a partially differentiated progeny that matures into an intermediate progenitor cell, which is a transit amplifying population that then divides to give rise to neurons. And so what's particularly appealing about this system, furthermore, is it's highly quantitative. There are exactly 8 type 2 neuroblasts per larval brain lobe. And so using our GFP tag Zelda, we could identify Zelda expression specifically in the neuroblasts and could show that it was rapidly eliminated following asymmetric division. So we performed ChIP-seq on larval brains enriched for these type 2 neuroblasts and identified 12,000 Zelda binding sites. And we could now overlay these binding sites with our ChIP-seq and identify 4,000 regions that are shared between the larval brain and the embryo. And this is what we might predict for a pioneer factor. But we identified thousands more that were specific to each tissue type. And furthermore, those regions where Zelda was bound specifically in the neuroblast were enriched for accessible chromatin. And so I'm summarizing a lot of our analysis, but we found that these shared peaks were enriched for the Zelda motif. But in the neuroblast, Zelda binding is not primarily sequence driven. In fact, in the neuroblast, it's binding to these regions that are accessible and do not have a canonical Zelda binding motif. And so here I'm showing a single example where we see binding in the embryo and accessibility in the embryo, and binding in the neuroblast and accessibility in the neuroblast. And this, in fact, was an enhancer that we could show for this gene tailless, and we could show that this neuroblast-specific binding was identifying neuroblast-specific enhancers. We could then specifically ask whether Zelda what happened when we overexpress Zelda, and we could overexpress Zelda in the type 2 neuroblasts and quantitate the number of neural stem cells. And we identified many more than 8 neural stem cells, showing that Zelda was promoting the stem cell fate. But this could either because it could suppress the asymmetric division or because it was reprogramming cells. So we then expressed Zelda in these intermediate progenitors in which it's not normally expressed. And we could again identify extraneural stem cells. And so what this told us was that Zelda could reprogram these partially differentiated progeny back to that undifferentiated fate. But when we went further out the lineage and expressed Zelda in the INPs, we now no longer saw extraneural stem cells. And so what this told us was that differentiation state was limiting this capacity of this pioneer factor to reprogram. And so here I'm again showing you this tailless enhancer that's accessible and bound in the neuroblast. And we could take advantage of a system that Cheng-Yu Lee's lab developed to synchronously essentially mimic this differentiation and look at what happens to chromatin accessibility. And what we found at this locus, but at many others, was that accessibility was specifically lost as these cells differentiated, suggesting that something was coming up and preventing a barrier to the ability of this pioneer factor to bind. And so using this system, we've now identified that there are barriers to this pioneer factor activity, and we have some thoughts about what that may be, including histone deacetylation. And so this is really important because we know that misexpression of pioneer factors in various tissues can lead to disease. So I've just told you that misexpression of ZELDA can lead to a neural stem cell like phenotype, tumor-like phenotype. Misexpression of DUX can lead in muscle cells, leads to FSHD. Misexpression of a pioneer factor grainy head that Megan from my lab talked about can lead to many cancers, as well as OCT4, SOX2, and KLF. And so what we really want to go now on to understand then is to use these two systems that we have, this blastoderm embryo in which multiple pioneer factors are cooperating, as well as these neural stem cells in which we've shown this sort of counterbalance between pioneer factor activity and differentiation, to understand how do pioneer factors cooperate to drive these reprogramming events? What are the unique properties of these factors that really allow them to be so act at the top of these regulatory networks? And then also identify the molecular barriers so that we can better understand how misexpression can lead to diseases. So with that, I will thank the people, the wonderful people in my lab who did this work, amazing collaborators, and our funding sources.
Hello, great talk. I was a bit confused with the degradative Phi experiment that you did. It's a technical question, but you expressed degradative Phi from zygotic genome.
So we use the nanos promoter. So it also is active in the germline as well, but it clearly is allowing embryos to get produced. Yeah.
Oh, I see. So that was a lucky thing that, you know, because germline clones are
dead.
But
so we know that it can make embryos. Yeah.
Oh, okay. Perfect. Thank you very much.
Yeah.
Well done. So, so you think that there's a blocking activity, I guess, for those neuroblast specific ZELDA sites. Is there any kind of an affinity difference that could contribute to that as well? So if you overexpress ZELDA in the embryo, do you start to fill those sites eventually?
Yeah, so we actually have some very preliminary data that suggests that there's a domain outside of the DNA binding domain that may be contributing to this, but it's really preliminary, so I can't speak molecularly to that at all. And we do know that if we overexpress ZELDA in the neuroblast and then look at binding, it can now occupy more sites, and many of these are CAG-TAG enriched. So we're trying to understand exactly that. Yeah.
Hey, Melissa, you might have covered this, but those different pioneering factors in the embryo, are they doing mutually exclusive gene sets or overlapping, or so you need them all because to get all the genes on, or what's the overlap there and what they're doing?
Yeah, so that's, I mean, it's complicated. So, I mean, of course, it's biology. So, I mean, it's not exactly, they bind to a lot of the same sets. We could show that GAF and ZELDA do not depend on each other for binding. But when we get rid of either one, we haven't gotten rid of both. We see different subsets that depend on them, but they could be complementing each other at different sites. So, yeah.
Okay, let's just do one more
Okay.
question and then we'll transition. So last one.
So maybe this is already known, but if ZELDA has to come off of the DNA with every mitotic cycle, then it has to come back on. Then it has to recruit other transcription factors and chromatin modifying factors. Does that mean that the timing of the mitotic cycle is a control mechanism for how many genes can be expressed? Because it just takes time to rebuild.
That's a great question. And I think this is slightly addressing it. So what we used, Liz had a poster in which she showed, Liz and Zoe, in which we looked at translation, and Zelda is precociously translated in a BRAT mutant. So this was building on work from Craig Smeeber and Howard Lipschitz. And so what we looked at was single embryo RNA-seq is what happens to genes. Are they precociously transcribed? And they're not. They're robustly active. They're overactivated at nuclear cycle 14, but we think that it's the nuclear cycle, as well as potentially the binding of these additional transcription factors that's limiting the ability to cell to activate. So I think that sort of gets at your question.
All right, let's thank
Thanks.
Melissa.
Okay, I can drink.
All right, well, I want to thank you all for sticking around and welcome Bob Johnston.
Well, thank you to the organizers, and thank you for everybody for sticking around. It is, I was nervous this morning, but then seeing these awesome talks in front of me, now I'm really freaking out. So fingers crossed, fingers crossed. All right, so a big question in biology for a long time has been, how do we get the different kinds of neurons in our nervous system? And some of the earliest, most beautiful work came from the tracings of Cajal. And the big picture question in the field is, how is neuronal diversity generated? And so often in development, we go to a kind of, I'm sure everybody's tired of seeing this kind of thing, but we go to this Waddington landscape where it's kind of like a marble going down a hill, and these cells take on different fates. And normally when we think about this, we think that inputs like signaling will push a cell into a certain fate, or lineage might push it into another fate. And ultimately, individuals that are genetically identical will appear almost identical to us. But there are some cases during development where this doesn't happen. And this is what we're interested in my lab. So in these cases, the cell goes down a path and it hits a kind of a choice point and it randomly decides. So an example in humans is we look in their eyes and we look at the red, green, and blue cone cells, we can see dramatically different patterns across individuals. And so what my lab focuses on is, how do cells make these random choices? And so the first question we ask is, well, how can we even study stochastic fate specification? And for us, we turn to everybody here's favorite model system, the fruit fly. And in particular, we study this in the context of the fly eye. And everybody in this room has looked at a fly at some point, hopefully. Maybe not the PIs. So, and you may be thinking, like, underlying this uniform morphology, there are actually two subtypes of R7 photoreceptors defined by expression of light detecting rhodopsin proteins, rhodopsin 4 in red here, and rhodopsin 3 in blue. And we dissect the eye and stain for these different opsins. We see this beautiful, amazing random pattern where each cell randomly chooses to be either the red fate or the blue. And it's not a perfect coin flip. It's about 2/3 of the cells choose to be red, about 1/3 choose to be blue. And don't overthink it. Just think of it like you're rolling the dice. 1 through 4, you get red. 5 and 6, you get blue. Okay. So the next question is, of course, well, what controls this decision? So we've known for a while now that a transcription factor called spineless, abbreviated SS, is expressed in a random subset of R7 photoreceptors. And if you're going to take one note during my talk, this is a good place for one, which is that spineless, in the cells that express spineless, you get expression of red rhodopsin 4. In the cells that don't express spineless, you get expression of blue rhodopsin 3. And throughout my talk, just for technical reasons, when we look in adult stages, we use rhodopsin 3 and 4 as a proxy to look at spineless expression. So this is great. We have this transcription factor, but we're just stepping up the chain, right? Well, what controls spineless? And today, what I'm going to tell you about, the main part of my talk is going to be about this antagonism between transcription and chromatin that controls the expression of this gene. Then I'm going to talk about the source of variability that ultimately decides which way it's going to choose. And then finally, some outstanding questions. So the main part of my talk today is work by two awesome grad students, Luke Bortman and Katie Anderson. And I've been telling you about the adult fruit fly eye. To get at this, they had to kind of go back in time and go into the larvae. And what I'm showing you in this picture, which you probably can't see well, but don't worry, I'm going to go through it in more detail, is we did RNA fish against the introns of spineless, so we can look at the nascent transcription of spineless. So each dot is what you're looking at is a nascent spot of transcription. So what do we see? Well, just to schematize this to make it a little easier to see, the eye antennal disc, which where we're studying right now, spineless is expressed in both regions. In the antenna, it's expressed in all the cells of the central part of the antenna, acting as a really great positive control for almost all of our experiments. In the eye, it's much more dynamic. The cells in the eye go through 4 main stages that I'm going to briefly tell you about. And what I'm going to do is I'm going to box some cells because when I'm about to show you, it looks like the cells are moving, but in fact, they're going through each of these 4 states. So initially, they're undifferentiated and spineless is off. As time goes on, there's this progression that moves from the posterior to the anterior, and every single cell will now express will go through a precursor state in which they all express spineless. As time moves on, these same cells will then turn off spineless as they differentiate. And then finally, as they mature into terminal R7 cells, they will choose to either randomly express spineless or not. And so the beautiful thing about this system is when we take individual eye discs, we can see all 4 stages that these cells will go through. The off undifferentiated state, the on precursor state, the off differentiating state, and then the randomly on or off R7 state. And this kind of explains R in situ here. All right, so I told you about the expression. One more important thing about this is that in the antenna, we see expression of both RNA and protein. In the precursors, we only see RNA. And in the R7s, we see, again, both RNA and protein, suggesting that any role for the expression of these precursors likely, I want to show you, is driven by transcription itself. So to get to the mechanism, the first question we ask is, what regulatory DNA elements control spineless expression? To get at this, we wanted to really hammer on the 60 kb locus. So we do what we call a reverse promoter bashing approach, in which we use CRISPR to knock out most of the non-coding DNA elements. And we found 3 key sites. So again, in wild type flies, about 2/3 of the cells will express spineless as indicated in red here. And we knock out each of these elements, we see a complete loss. Now, one's the promoter, kind of boring, but the other 2 suggest that these might be enhancers. So what I'm going to show you now is that we name these the early enhancer and the late enhancer. And when we hooked up the first element to a reporter, what we saw is that this early enhancer drove expression early in the precursors, whereas the late enhancer drove expression in all R7s in the terminally differentiating cells. All right, so now we kind of have these enhancer elements. We wanted to dig in a bit deeper as what is their role in this process. So the next question we ask is, what is the role of the late enhancer? And to get at this, what I'm going to show is the kind of 3 main areas in which spineless is expressed. In the central antenna, where spineless is expressed in all the cells, in the precursors, where it's again expressed in all the cells, and then finally in adults, as indicated in red here by rhodopsin 4. So we said, okay, let's knock out this late enhancer. What do we see? Well, in the antenna, we still see beautiful expression. In the precursors, again, we see expression. And then in the late stage, we see a complete loss, consistent with this enhancer playing a specific role to drive expression late in development. So that seems pretty obvious. So the late enhancer is required for expression in R7s. The next thing we wondered is, what is the role of the early enhancer? And this is where things started to get interesting. So in the antenna, we knocked out the early enhancer, and in the antenna, we still saw expression. In the precursors, where we know that this is able to drive expression, we see a complete loss of expression again. But interestingly, we also see a complete loss in the terminal R7s. So what this suggested to us is that this early enhancer is required for both early and late expression. So the next question we wondered is, it seemed like there was a relationship between this early and late expression, and we wondered, could we tune it? This is pretty blunt force things. We're just knocking out enhancers, right? Could we kind of turn it down and see what happened, or turn it up? So The first thing we did is we said, okay, what if we just kind of turn down expression early? Will we see a decrease in the number of cells that express late? And so we previously identified a cis regulatory change, a single base pair change in the early enhancer. And what we see here is indeed in this change, we see a decrease in the expression in the precursor stage, and we also see a decrease in the percent of cells that express late. Great, so we can tune it down. We now see a change late. Of course, we wanted to ask the opposite question is what if we now de-repress it early, allow more expression? And in that same paper, we found that CLU repressor acts on this region. And so we knocked this out. So there haven't been too many clones at this meeting. So all the old timers will appreciate this, and maybe the new kids are like, what? So I'm gonna briefly go through this. This is a clone. I think it's a beautiful experiment though, so I have to share it. So in purple, what you're seeing is wild type tissue. In the non-purple cells, you're seeing CLU mutant. And first I wanna highlight the precursor stage. We see no change between the mutant and the wild type tissue. What was interesting is the next stage, the differentiating cells, we saw a difference. So as I mentioned before, in wild type tissue, spineless turns off, but in CLU mutant tissue, we see that spineless remains on, suggesting that we've now extended expression from the second stage to the third stage. So what happens? Well, ultimately in adults, we also see this increase in the ratio of cells that express spineless. So what I've shown you is that, okay, great, this early expression controls this late expression. So that's cool, but of course, as Melissa eloquently said, it's biology, it's more complicated. That's the case. So as I mentioned earlier, spineless is expressed in a random subset of cells, but this late enhancer drives expression in all the cells. So we are missing a repression step somewhere late. And so the next big question we want to ask is what turns off late expression? And a big question is how can we even test for repression in R7s? So we wanted to do what we call a repression reporter. Now, enhancer reporters are very common to look at can a region activate expression. We wanted to see if the locus could repress expression. So to get at this, we took a kind of broad eye enhancer and hooked it up to a reporter, and we inserted this reporter into the spineless locus using CRISPR. In other loci, this enhancer drives expression in all the cells. And we had two main hypotheses. One is, if there is no repression occurring in the spineless locus, we should still see this reporter in all the cells. But if the spineless locus was sufficient to turn off expression, we should see it now limited to a subset of cells. So we did this experiment, and so let me just show you the controls. Again, we have our spineless on red cells, our spineless off blue cells, and our reporter, this reporter is expressed in all R7s, and in fact, all photoreceptors of the eye. So now we put it into spineless, and what do we see? Well, we see that this reporter expression is restricted to only the spineless on R7s. So it's turned off in the spineless off cells, including our spineless off R7s and outer photoreceptors. All right, so this was pretty cool. But of course, this is one site. Maybe we just hit some funky site in spineless, yada, yada. We wondered what could we learn by putting this into other sites? So we did similar experiments where we now put it into 5 different sites, and we saw the same effect. The repression limited expression to a subset of R7s. And in fact, 4 of these sites were in the transcribed region, but one was even outside of that transcribed region. Together, this suggested that repression of spineless affects the entire locus. And it really pointed us in a direction that chromatin's playing a role. So the next big question we asked is, well, how does spineless chromatin compaction change during development? And there are a lot of challenges. One is it's very heterogeneous tissue, and we really wanted to look at this in a very kind of refined cell type specific way. So to get at this, we turned to a visual approach. And our idea here was, let's look at the spineless locus and let's use DNA fish. So we labeled the upstream region in blue here, the spineless locus in green, and the downstream region in red. And what do we see? Well, in our controls in spineless off cells, hopefully you can appreciate it, that this is more compact. And in the on cells, it's more open. And to quantitate this, we took the center of each of these dots and measured the distance between the dots. And so this is kind of just a toy schematic here, but ultimately we could add up these distances to get an overall quantitative score for our compaction rate. So we then took this 3-color DNA fish assay and said, okay, how is compaction changing over development? And indeed, what we found is that it's dynamic. So what I'm going to show you is I'm going to kind of walk through the different key time points in this process. So first, as our negative control, we use what's called the peripyloid membrane, which ensheaths this tissue, and it's not on the schematic here, but spinous is off. So this kind of gave us our baseline off compaction rate. We then went to our nice control of the antenna, which spinous is on, and we'd see this kind of pretty significant opening of the chromatin. Great, so we have kind of our off compact and our on open controls. What happens in the eye? So in differentiating cells that are off, we see that this is compact. And what was really exciting to us is it's very reproducible to this other cell type. What happens in the precursors, which all the cells are on? Now these are all open. Great. Now the next question is spinous then turns off. So what do you think is going to happen? Well, that's not what's going to happen. So what we see is that we see this intermediate compaction rate. And we had two main hypotheses at this point. One is perhaps this is just a big jumbled mess. You know, the chromatin in all different kind of conformations. Or perhaps that there's an open and closed population and we just can't see it yet. And in fact, this intermediate compaction rate was maintained in our 7 cells, which is a mix. Well, this is all our 7 cells, but this is a mix of both on and off cells. So to get at this, this is actually a harder experiment than one might think, but we had to figure out a way to do this with a reporter. And what we did then was we said, okay, let's break it down and look specifically in the on and off our 7 cells. So we did this, and indeed, in the on cells, the chromatin was more open, and in the off cells, it was more compact. So together at this point, what we had is kind of this main model here, which is that during the specification of these R7 photoreceptors, initially spinous is off and compact, then spinous is turned on and opens all the loci. It then turns off and either compacts or remains open. It maintains this state until this late enhancer turns on. In the cells that are open, it is now able to express, and the cells where it's off, it now turns off. Sorry, when it's compact, it turns off. All right, so this is a great model, but we need some experiments to actually prove this, right? So what do we do? You know, I ask people a lot when I talk about this work, you know, if I had to ask you if transcription controls chromatin or chromatin controls transcription, what would you say? And all of your answers are right. So we basically, that's what this came down to, is which is controlling which. So the first thing we did is we knocked out the promoter. So we completely lose expression in all cell types. And in all cell types, we saw that it was compact. And what this suggested to us was that chromatin opening required the promoter and transcription. The next question we said is, does early expression specifically open chromatin? And to get at this, we had our nice tool set here with the CRISPR mutants. So we looked at our early enhancer mutant. And again, we had our controls. So our off control was compact. Our on antennal cells were open. What happens in the eye? Well, we see that it never opens. So again, what this argues is that you need transcription driven by this early enhancer to open the chromatin moving forward. Okay, so early transcription opens chromatin. The next question we asked is, does late expression affect compaction? And so to get at this, we again went to our knockout, and we went to our negative control that's compact, our on cells that are open, and we looked into the eye. In their first 3 states, we expected no effect, and we didn't see any effect. In other words, the undifferentiated cells were closed and off, the precursors were on and open, and then the differentiating cells were this intermediate. In the R7s, you could have 2 main predictions. You now lost this activating input. Either they could close, or perhaps they would maintain intermediate. And in fact, they did remain intermediate. And so what this tells us is that the compaction in R7s is independent of that late enhancer. So I've told you a lot of data. Let me summarize what we're talking about here. So this is just a fancy word for the title of our paper. But there's just a dynamic, I don't know, dynamic always seems like me, it should be on the second scale, and this is like on the hours, I don't know. There's this dynamic antagonism where early transcription drives chromatin decompaction, and late in this process, chromatin compaction represses transcription. And so just to get a little more detail in this model, what we think is happening is that early, the spinous locus is in a compact state and off. Then the early enhancer drives expression to open the locus. It then turns off and the locus either remains compact or, compacts or remains open. And then finally, repression limits expression driven by the late enhancer. So at this point, what's the big picture here? So stochastic fate specification is a bit of a niche field, so there's not a lot of understanding of how this works. But in the two other well-known models, in bacteria, there's a lot of variable gene expression, which ultimately triggers a stable cell fate. And so that kind of resembles our pulse of early expression. And then in the mouse olfactory system, it's well known that repression via compaction plays a role. And we think that in the fly eye, this is kind of bringing together those two principles. So, all right, so I've kind of outlined this mechanism, but I didn't actually tell you what's controlling the decision, which is kind of the biggest question in this field. And we're working on that. So this is ongoing work getting at this source of variability. And this is work by Liz Urban, an awesome grad student in the lab. So kind of going through this mechanism, we can really focus in on this step. What makes this chromatin decide to be closed or open ultimately? Is it variability in the transcription, the chromatin compaction, or both? And so to hammer on that, Liz went to town. And so what Liz did is she conducted, I've been telling you about just one color RNA fish. She decided to do 3 color RNA fish, labeling the 5' end in red, the middle in blue, and the 3' in green. And what this told us roughly is when this gene is being initiated, elongated, and terminated. And that kind of is right here. Cells that are just starting to transcribe will be more red, continuing to be red and blue. And if they're kind of continuously expressed, they'll have red, green, and blue. So what do we see? So in the antenna that's continuously expressed, we saw kind of continuous expression, meaning like hopefully you can see here that all the dots in the merge are white, meaning that we're getting constant initiation, elongation, and termination. What about in the precursors? So in the precursors, it's variable. So we're zoomed in here, and let me just show you 3 examples. First off, hopefully in the merge, you can see it's all different kinds of colors. That's a good thing, at least it's exciting to us, because that suggests variability. And if we look, there's some cells that have all 3, suggesting they're have multiple transcription events going on. There are cells that have just the 5' in middle, suggesting they're basically just elongating at that point. And then cells that only have the 3', hopefully you can see that in green there, suggesting these are terminating. I just want to emphasize these are intronic probes, so likely the 5' in middle are being spliced out. All right, great. So there's variability, but what is the nature of this variability? So transcription is sometimes a continuous process, but often it's susceptible to variability and bursting. And so, we wanted to get at how this could be working. So to get at this, what Liz did is she compared different regions to see what states relative to one another in. And normally, if you have continuous expression, you'd expect the ratio of these 2 signals to be almost the same. So you'd see a nice peak here. If there were more cells, this would be our continuous steady state model. If there are more cells that were initiating, this would then be shifted to the right. If more cells were terminating, this would be shifted to the left. And then if it was just broadly more noisy, more just variable, you would see kind of this peak being further spread out. And this was our prediction, is that there should be a spread in the distribution in precursors. So Liz did these experiments and she compared all the combos, and what she saw is indeed that. So we see a nice tight peak for the antenna, but we can see, hopefully you can see there that the precursors are more spread out. When we compare the 5' to the middle, we see the same thing when we compare the 5' to the 3'. And finally, we see the same thing when we compare the middle to the 3'. So what this argued is that spineless expression is variable in precursors. But the next thing Liz wanted to know is, is it just like something like developmental timing that's explaining this? The antenna is just continuous, but I just said that, okay, this goes through all these different states, in time. And so to get at this, Liz teamed up with Luke and said, okay, let's look at where these, the different states are relative to development, focusing on the fact that the posterior cells will be the oldest and the anterior cells will be the youngest. They fit these positions to a curve, and then they measured the positions relative to this curve to get at the developmental timing. So we had 2 predictions. One is that if developmental timing explains the variability, we should see a curve like this, where as the intensity would increase as spineless turns on in the youngest cells, and then decreases in the cells that are the oldest as spineless turns off. On the flip side, if developmental timing didn't explain it, it should be just kind of this random fun mess. And indeed, this is what we saw, suggesting that the expression in these precursor cells is variable across the board. So Liz has really definitively shown that expression in these precursor cells is variable. And now the last challenge she's fighting to do is kind of link this to the decision. So what I've shown you is it's variable in precursors. And what I showed you in the first part is that the early expression ultimately dictates the late expression. So what she's working to do now is determine how does variable transcription influence these fates. And to get at this, she's using an MS2 system where we insert hairpins that are recognized by the MCP mCherry. And when spineless is transcribed, you'd expect that you get binding by this MCP mCherry, such that when spineless is on, you'd see a red dot, or when spineless is off, you'd see no dot. And so these are ongoing experiments, but I'll show you these awesome movies that she's working on now. In the antenna, where we see continuous expression, just focus mainly on 2 cells on the far left. What you can see is that we see expression. And to get at this, what Liz had to do was dissect out the fly eye antennal disc, mount it onto the confocal microscope, and keep it alive for many, many hours. So this is really fantastic work. But this is fine. What do we care about? It's the precursor cells. First, hopefully you can see this kind of streak. It's a little bit on the diagonal there, but you can see this line of precursor cells, and we have this working in these cells. So I wish I had the final conclusion, but this is just our ongoing work. And what Liz is doing now is she is now working to connect the noise and transcription to ultimately this R7 fate decision. And we're currently trying to recruit people to work on the next big question, which is to develop a live system where we connect this live transcription system to a live chromatin system, which we're working on in the lab. All right, so I'll just conclude with a couple outstanding questions. One is, how are stochastic and stereotype patterning coordinated? You look at the fly eye, it's very uniform. I just told you there's this randomness underlying it. Well, Ali Ordway, an awesome postdoc in the lab, has been working on this problem. And what she found is that hedgehog plays a key role. Normally, hedgehog is at its normal levels. You see this beautiful eye and about 67% spineless. As we decrease hedgehog, we decrease the size of the eye, and we decrease the ratio of the cells that express spineless. And this extends if we really kind of obliterate hedgehog activity. Other questions is, how general is this mechanism? And what we're trying to do, we have identified enhancers that could putatively respond in this way in genes, and we're trying to address this question. We're trying to get people in the lab to work on this question, is how does this work in the eye, but also we'd like to extend these studies to the embryo. And then finally, I started with this picture of Cajal, one of the big inspirations across the developmental neuroscience field. And we've extended our work beyond fruit flies to study these kind of questions, to ask the questions, how are the different types of neurons generated in the human eye? So to get at this, we take human stem cells and we differentiate these into what's called human retinal organoids. And using this system, we study how the red, green, and blue cone photoreceptors of the eye are made, and also how the retinal ganglion cells are made in the human eye. And with that, I thank the organizers once again. We are recruiting postdocs, and thank you, everyone.
So H2AV is supposedly alternate histone is incorporated into active transcription and interphase. I'm wondering if in your early expression, you're incorporating H2AV and that makes the locus more accessible later.
So that is a fantastic question. And we had that hypothesis for a long time. And it's still, so just to repeat the question, the idea is, could the transcription then be putting other histones in there that could be opening the chromatin? So we sort of addressed it in a different way. What we did is Liz used CRISPR to put a transcriptional stop very shortly after the initiation, hoping to see that exact effect, but we saw no effect. So it appears that you do not need, at least through the whole, it doesn't rule out that maybe the early part still could be playing a role there, but we wish it was that, to be honest with you. Erica.
Very exciting talk, Bob. That was beautiful.
Thank you.
I was just wondering about that enhancer and whether or not there's additional sequences in it that potentially, it might not be the transcription itself, it could be that it's recruiting some chromatin remodelers that might open the chromatin.
Yeah, so 100%. So this is kind of the question, is it the elements or is it transcription itself? And, we do think it's transcription because we knock out the promoter or the enhancer and we see this effect. It still doesn't rule out, it's a really hard question, right? Because how do you mess with the enhancers without messing with transcription? What I will say is this, is we see binding sites for your favorite clamp, and we've tested it previously, but we don't see any effect. But there's a lot of caveats with that because maybe with the eye, you always have to deal with perdurance issues and stuff like that. But it's definitely a question we want to revisit.
Thank you.
Thank you.
Hey, Bob. Oh,
Hey.
thank you for the clones. You officially made it a fly meeting. My question is, so spineless seems to have different isoforms that actually lead to different peptide sizes, right? And one way of really fine-tuning activity of genes is by making them slightly be more either stable or translated or slightly less efficient in function. So do you have any ideas or hints that if at these different stages, potentially the longer or the shorter isoforms may be more present, and would that play any roles here?
Yeah, so this is a great question. This kind of feeds back a little bit, at least in terms of the mechanism of expression. We don't think the isoforms are playing a role there because we put this early stop and we don't see an effect. But there's a bigger question, is like, we don't have any idea, and we'd love to work on it, is that early precursor stage, we don't get protein. And we have no idea. And the coolest hypothesis I've had for about, this is gonna make me sound old, but I've had for like 15 years now, when the original, I don't even know which paper it was, but it was one of the genomics papers. There's actually an exonic microRNA in spinous, an exonic microRNA. And that's like my favorite hypothesis of what's controlling it. But we just need somebody to come in and use CRISPR and then break up the hairpin and see what happens. But infinite time, infinite money, infinite people.
I'm sorry, in the interest of time, I know some people.
Oh, come
want
on, I'm
to
last. I've earned this.
So hopefully Bob will be around for a few minutes. So.
Thank you again to the organizers. Dave, we should have one more big round of applause for the organizers.
Thank you. Thank you so much. I'm losing my voice. Thank you so much for coming. Thank you to every single person who spoke here and attended meetings and took notes and just made this meeting a wonderful experience. And I also hope that you consider please coming to the next meeting in 2023. Let's not have a drop off in the next meeting, which I think will also be fabulous. So this is the end of DROS 2022. And I hope you all have a safe journey home. Thanks.
Oh, yay. Oh, yay, yay. Thank you. It was fabulous. Come to New York with your kids, please. Your son and Ishan can play video games and PS5 and all kinds of stuff. Yeah, come. I will. Thank you, Amy.